I would like to say a few words about our today's very special guest. We are very pleased to have Professor Marcin Kołaczkowski with us. Probably some of you know, some of you don't, that Professor Kołaczkowski is the Dean of the Faculty of Pharmacy at the Jagiellonian University Medical College and also heads the Department of Medicinal Chemistry. Professor Kołaczkowski has almost 25 years of experience as an academic researcher, and it's worth mentioning that he has made substantial contribution to medicinal and computational chemistry, and particularly GPCR targeted drug design, and specifically a topic that is very close to my heart, biased serotonin 1A agonists, an area of growing importance in modern neuropsychopharmacology. Moreover, for the last 18 years, which is very important, Professor Kołaczkowski has also served as the lead scientist for CNS programs at Adamet Pharma. Adamet Pharma is the second biggest Polish pharma company, so it's worth mentioning this fact, and his work exemplifies the rare ability to seamlessly transition between high-level academic science and real-world application. Honestly, a quality that is talked about very often, but achieved by few. Except of that, Professor Kołaczkowski has co-authored over 140 peer-reviewed publications and is also a co-inventor on 10 patents. It's quite a lot. And this really reflects his scientific productivity, but also his sustained focus on developing new therapeutic strategies that may one day shift clinical paradigms. I would also like to stress out that the programs he led were commercialized to pharma or biotech partners, which is very important. In recognition of his achievements, he was also awarded Roman Kalishan Award, a national distinction for outstanding contributions to pharmaceutical sciences in Poland. Except for the scientific part, there is also a other very important part, which is education. Professor Kołaczkowski is a very committed academic leader with a strong focus on education. In addition to teaching pharmacy students, he co-founded the Interdisciplinary Drug Discovery and Development Master's Program, which some of you here today attend. And you need to know that he supervised the design of a practice-oriented curriculum in this program that can prepare students for diverse career paths in the pharmaceutical industry. And this includes both innovative and generic drug development. I think it's very important, and it's the first such program in Poland, so it's worth mentioning. And as the Dean, Professor Kołaczkowski supports a research-driven, collaborative academic environment that encourages innovation and excellence, which is, I think, very important in our times nowadays. We are very fortunate to have Professor Kołaczkowski here today with us, and I really look forward to his seminar. So without further ado, I would like to give the floor to Professor Kołaczkowski.
Thank you very much, Karolina, for this really nice introduction. I haven't heard so much good stuff about myself for a long time, so I feel flattered. Besides all this you said about my success, I must say that it was also a series of drug discovery failures over this more than 15 years. I probably made most of the mistakes I will be talking about today. So I am not talking as a guru who knows more than others, but rather as a person who experienced these failures on my own skin. But I want to share it with everybody, maybe to spare some of the hurdles for the upcoming endeavors. Okay, so let me share the screen with you. Okay. Do you see my presentation?
Yes, we can.
Yes.
Okay. So let's start. When I was thinking about the topic for today, I asked this question: why all these brilliant molecules don't become drugs? We hear many times that the scientists came up with some great ideas, great molecules, and we also hear about some frustrations of scientists who cannot understand why their great achievements in science, in laboratory, do not translate to commercialized drugs that enter into therapy. I am not sure I know all the answers to these questions, but definitely some of them are most probably true. I would like to start with this quote of Dr. Mark Marco, a former chief technology officer from Vertex Pharmaceutical, one of the biggest innovative companies in the world. And he said that there really aren't any problems that are more complicated or more satisfying from a human perspective than producing novel medicine. Producing meaning bringing them from ideas to patients. Okay, so what he says, and he knows what he's saying because he succeeded and his team succeeded many times in bringing novel molecules to the market, that this is greatly satisfying, but also very complicated. And there is a term rocket science, and I would say that definitely bringing new molecules to the market is a rocket science. Okay, we want to answer why all these brilliant molecules do not become a drug. So let's first answer what is a drug and what does it take to be a drug. Drug may be defined in various ways. A drug is any substance that causes temporary physiological and often psychological change in the body, especially in English language. The term drug, which comes from French, by the way, means many different substances that exert a pharmacological effect on the body. Also street drugs, also narcotics, but these are not drugs that we want to talk about today. We want to talk about pharmaceutical drugs, also called medications or medicines. And such a drug is a chemical substance, which is very important. I also stress out this fact to my students, that drug is not a name. It's not a string of letters. It's not a spell or a goodwill. It is a chemical substance. This is why medicinal chemistry, which I deal with for almost 25 years, is so important for pharmacology, also in pharmacotherapy. This chemical substance is used to treat, cure, prevent, or diagnose a disease, or to promote a well-being. So we have a strictly medical context of this very well characterized chemical substance. But what is lacking in this generally applied definition? The answer is the regulatory aspect, and this is the first thing I would like to focus on today. A pharmaceutical drug is a chemical substance approved by the appropriate authority to treat, cure, prevent, or diagnose a disease. Okay, so if we had a substance that we think has some activity, or we even made some clinical trials, but it was not approved by the appropriate authority, this is not a drug. This is not a medicine. Okay, we may believe in its potential, but this is not a drug, and without this approval, it will never be. Neither will it be used for patient care, neither will it generate any profit to the company. So who are these authorities who decide if our brilliant molecule will be a drug or not? There are various agencies around the world. They are called regulatory agencies. They approve drugs, label them, monitor their use, especially safety, They can also withdraw a drug or change labeling, for example, add warnings, limit use, etc., which can be very important commercially also for a company. There are two key players, the American US FDA and European Medicines Agency, so Food and Drug Administration and European Medicines Agency in Europe, EMA. Why they are key players? I think FDA is the oldest and the more opinion leading, and EMA also covers a lot of the important European countries. So together they cover probably 80% of most important pharma markets all over the world. If you want to have a drug on the market, you have to satisfy them first. They are, therefore, your primary target. You won't go to doctors or patients or anybody else without passing them. Patients are our ultimate goal. For prescribed drugs, we cannot escape also prescribers, doctors. But this is not our target. Our target are the regulatory agencies. So we have to learn about them as much as possible to know our enemy. Drug discovery and development workflow is like you see. We have the preclinical phase, so we start from the idea, from in silico screening, from chemical synthesis, from in vitro studies using cell lines, other in vitro or ex vivo techniques, finally animals, and we have clinical part where the studies are executed with help of the patients. And between these two stages and at the end of the clinical part, we have two main regulatory milestones. First of them is called IND, so Innovative New Drug in the US, or CTA, Clinical Trial Application in the EU, while at the end of our journey, we have New Drug Application in the US and MAA, Market Authorization Approval, in the European Union. To get to the market, we have to have the final approval. To get to this final approval stage, we have to go through the clinical trials. To get there, we have to pass through IND. And for us, for the researchers at the preclinical stage, this is the first major milestone to be achieved if we want to turn our brilliant molecule into a drug, and we cannot forget about it. Here at the beginning of this journey, we start to test our compounds, design them, synthesize, and test in vitro, then in vivo. Here at the end of the lead optimization stage, we select our compound, the compound, the one that we select here lives or dies throughout the other way. So this hit to lead and lead optimization stage decides what happens later. The part of our journey during which we select this molecule is called drug discovery. The part of the journey during which we characterize this molecule and develop also the drug form, drug product formulation, etc., is called drug development. So drug discovery is obtaining and testing many substances to select a drug candidate. Development, these are studies of the selected drug candidate required for approval. Sometimes drug discovery workflow is showed as a funnel. Funnel starting with many molecules which go down to one. And if you see here, for example, the in vivo efficacy assays, which are very important proof of concept at the preclinical stage, are only one of many stages that lead us through this journey. So in drug discovery, we test many substances. We iteratively repeat design, synthesis, testing to optimize the properties, pharmacodynamics, pharmacokinetics, patentability, feasibility of synthesis and formulation, and we select one compound whose structure will not change anymore. Here, a potential drug is born. And will last till the end of the project for good or for bad. In drug development, we are testing this one compound. We are performing studies which are required by the regulators, preclinical and clinical. We develop a drug product, and what we do also, we cannot forget, we complete the documentation, allowing for approval, first for clinical trials, and then for hopefully final approval. So these are two phases which together form a whole process. What is the cost and the success rate of drug discovery and development? First, I said about the regulatory context, which cannot be forgotten. Now I want to talk about another problem, money. Okay, if you look at these stages, each of them has a success rate. Success rate can be compared to the yield of chemical reactions. So for all my colleagues who have ever done any synthesis during their work or during chemical classes and calculated the overall chemical yield, know that even if the yields of certain stages are quite high, the overall yield will be quite low if we have so many stages, each of them taking out something. We see the preclinical phase here, we see clinical phases here, and regulatory phase at the end. These phase 2 clinical trials are called the Valley of Death. The success rate here is about 34%. So 1/3 only of the molecules that come to phase 2 are translated to phase 3. No other stage is so killing the molecules. What is this stage? This stage tells us if the molecule that we have selected here during preclinical studies are really working in ill patients. Okay, so the low success rate here is because of poor translatability of the preclinical studies. And unfortunately, the poor translatability is especially poor in the case of the CNS programs, especially those which deal with some higher processes like memory, like mood, and also some neurodegeneration programs, which are a graveyard here. So what to remember from this slide? There is maybe a 4% chance that when we start a program, we will finish successfully. Not very convincing, not very encouraging, right? And second one, that the Valley of Death is in phase 2. In 2011, it was estimated that a single new drug cost about $2 billion to discover and develop throughout the process, most of which, of course, are clinical trials, but not only. Let's say that today it's about $2 billion. What can we do for this money instead of developing a new drug? We can buy 17 Boeing 347 aircrafts. We can build or buy 7,000 homes or 70,000 cars, or we can raise 7,000 children to the age of 18. Why am I saying that? I am saying that because it shows what else can be done instead of investing huge money into drug discovery and development. The investors not only look for profit, they also take under consideration the risk. And the risk in drug discovery and development is much higher than in these cases. We can build our own airline or fleet of cars around the world and earn some money much more convincingly than investing in drug development. So what's the conclusion for us based on that? If someone is to spend so much money on such a risky business, they really must feel it pays off. So in our project, we have to show them the money at the end, the value of our project. If we don't do that, they will spend this money on something else. Let's take a quick look at the life cycle of a pharmaceutical drug. I'm sometimes, laughing inside when I am hearing the theories that the company is keeping something in a drawer or killing some programs or doesn't allow some good molecules to be developed. This is not true. And the reason for that is the life cycle of a pharmaceutical drug. Let's take a look at it together for a moment. We have a preclinical phase here. Do you see my laser pointer?
Yes, yes.
Okay.
Yes.
In this preclinical phase, we are here, okay? Here, from the beginning to patent usually, maybe a little bit less. So you see that us scientists, we are only at the very beginning of the journey. Maybe in time it is not so short, but in terms of money that go around us, it is a little bit, okay? Then we have preclinical development, which is usually done in GLP standard. If we enter clinical trials, we have phase 1, phase 2, phase 3, and we are spending money all the time. We do not earn anything, but we are spending a lot. Area under this curve shows millions of dollars, hundreds of millions of dollars. If we are lucky enough to register our molecule to have an approval, we start to earn money. But if we start to earn money, first of all, we are not starting to earn a lot at the very, very start. It has to grow. But let's say our drug is selling well. We are still not on plus in our financial balance. After about 5 years or so of good sales, we are able to break even. So be on zero with the money that we spent. 5 years after approval. Depends on the project, of course, sometimes less, sometimes more. And then we start to earn money, okay? We earn money, we earn a lot, and then there is a decline. The competition appears. Our prices erode by 80%. This is a generic competition. Why they have appeared? Because our patent went off. The patent that we made, for example, recently, when we discovered, an interesting group of compounds here at academia or in a small company or in a bigger company, and we want to do something with development, we should patent, okay? It takes 20 years to protect. But the drug development, as you heard 10 minutes ago, lasts about 15. So we have probably 5 to 10 years of market exclusivity. If our drug sells well, we may break even or even earn on it. If not, because it sells not very well, or the patent was older, or it was somehow broken by the competition, we may not earn anything or earn just a few. Moreover, remember that we are not only earning the money to pay for what we spent on this program, we have to also pay for the 23 or 24 programs that went wrong on this whole DDD workflow, okay? Because 1 in 25 gets to the market, and all the other also cost, not all the sum, because they fail somewhere on the way. This is why we say fail fast. If you are going to fail, fail fast. The worst case scenario is failing in phase 3, okay? But anyway, the drug that entered the market has to earn for all the rest. Why is it important for us? Because if we are preparing to have a drug candidate, we have to have it in mind that when we are talking with investors, they have this picture in their mind, and they analyze how our proposal fits to this. If our patent is too old or non-existent, nothing will happen. Everything is regulatory driven, but don't forget the market. Why is market so important? Because it gives or doesn't give money. Money is needed to develop your product, more or less, but always quite a lot. Money is a reason for your investors or owners of the company for having this drug, for having this project in portfolio. In the end, most or almost every management decision is made in the financial context. This is called a value-driven management. Your product or project has to be financially attractive. It has to bear a promise of a high benefit to cost times risk ratio. Okay, this is the clue. Your program, when you are designing it, when you are thinking of what molecule you are bringing to the market, has to promise a huge benefit for the patients, benefit for the company, because it has to overcome the cost and the risk of the project. Cost is huge and risk is also huge. So the benefit must be huge. And your project, if successful, has to earn for the other failed programs. To secure it, you have to have a sufficient market exclusivity, which requires IP protection. So this is why patenting is so important. If you don't have this, there will be no drug on the market. What you have to consider when you approach a DDD project, first of all, is a medical need. A real medical need, well-established, consulted with clinicians, medical need. Not something that you read in the review paper of your colleagues from different city, but a real medical need. You need a science to convince yourself and your partners, the investors, that you have an idea how to address this unmet medical need. But that's not all. You have to have people, technology, infrastructure, so your project has to be doable in both infrastructure and people who know how to use the technology. Not only idea, but you have to be able to execute it. Patent or other IP protection has to be there. You have to have a clear idea of how to achieve it, how to achieve your protection. from the very beginning. You have to estimate the market size. The bigger patient potential, the bigger money pot at the end, the better. If it's small, your chances are smaller because the cost is usually always big. So the bigger the reward, the better for you. Of course, if you are looking for rare diseases, there are also foundations and patients, organization, etc., governmental money, which can help. But this is a niche. The let's say real world normal drug development is aimed at the general population, and the market size is very important. Another very important aspect is competition, because you are not alone. If it's an unmet need, a real unmet need, and the science is probably not something that only you know on this world, you would probably have a competition. And this competition may be better than you, they may be more experienced, they may have more money, they may have started earlier. So there's a question if it makes any sense to compete with them. You have to, first of all, know about their existence and know at which stage they are. And then find a way to convince the others that you will be better than them. And last but not least, money. You have to find money. If you won't have it, there will be no project. If you work in a huge company, this company has money, but it doesn't mean that it will give it to your project. So even if you are a big company, you compete for the money of this company. If you are in a smaller company, you most probably will have to either enter into alliance with a bigger company or take money from the stock market. And the investors can invest in anything. In something apart, pharma project, or in other pharma projects, maybe from completely different area. The bigger the market, the stronger the patent, the lower the competition, the more probable is that you will get the money. The first stage of having new drug candidate is having a validated hit, so a compound that works sufficiently at our target. Then a very important, a pivotal, crucial stage is having a proper lead structure. And last but not least, having a candidate. If you look at what a lead structure is, it is a promising combination of various features. So effectiveness, of course, but also selectivity, physicochemical properties, and early ADME tox results, and patentability, which are not yet optimal, but have some space for improvement. And this improvement is made in the lead optimization stage, after which you select your candidate. And the candidate has to have a satisfying, again, combination of features. Again, effectiveness, but also selectivity. We know that. Physicochemical properties translating to good pharmacokinetics and toxicological properties. Patentability, also synthesis scalability. And you have to show advantage over competition in everything, or at least having good properties overall and some advantage in efficacy or safety. Very strictly shown to convince that it is worth risking all this money to your project. After you assess all this stuff, you have to enter a stage which is called a preclinical development. So it's already a development because it's done on your selected drug candidate, but it's done before the clinics. And this is a stage which is overlooked by many. And this is a big mistake because this is a huge milestone in this whole DDD process. What we have to do at this stage? We have to do something which is called CMC, Chemistry Manufacturing Controls. We have to produce and analyze 2 batches of our compound, tox batch, so the batch that will be used in toxicology studies, and the clinical batch, so a batch of a compound that will be used in at least first stages of clinical trials. The problem is that these are not milligrams or grams. The problem is that these are kilograms. And another problem is that clinical batch has to be made in GMP, and tox batch has to be made maybe not in GMP, but also has to be quite clean because it will be used in toxicological studies. But that's not it. Not all. You have to develop bioanalytical methods and validate them in non-GLP standard and GLP standard, because you will have to assess the concentration of your compound in the various body fluids. And this is also very important and mostly overlooked stage. Then we have toxicology in rodents, exploratory, and the pivotal GLP study. And toxicology studies in non-rodents, again, exploratory and pivotal, longer, 14 or 28 days studies using dogs or monkeys. If that's okay, good, but it's only halfway. You have to do safety pharmacology studies, including respiratory system, CNS, and circulation, okay, in which you test not very high doses of a drug, some doses which are considered therapeutic, to seek for specific side effects which are most common. And this can be killer of your project also, even if tox went well. Another stage is genotoxicity studies, Ames test, micronucleus test, chromosomal aberration, etc. in GLP. Extended pharmacokinetics is also maybe not required, but very important to see the mass balance, this fuel distribution of your compound. And then at the end, you have to show the intended protocol for clinical trials. All this takes at least 2 years and costs between $3 and $5 million for a small molecule and much more for a biological drug candidate. This cannot be done by academia and cannot be done by a small company. This requires, or can be done, but requires significant, at least $3 to $5 million investment. So this is the first stage of development which we have to do with somebody else, with our partner or with the investment after we make a spin-off company or something like that. Optimization is always multi-parametric. We have effectiveness, but we have also selectivity. We have solubility, permeability, metabolic stability. We have patentability. We have to have clean structural alerts, so we cannot have any toxicophores in the molecule, etc. And the molecule that is selected as a drug candidate is not the most potent, not the safest, not the best absorbed, not the most soluble, not the most stable, but it's the best combination of all. But when we are optimizing these properties, there is a short blanket syndrome. We are optimizing safety, we are losing physicochemical properties, we are increasing solubility, we can lose effectiveness, or etc. So this is very problematic. And remember, unfortunately, potency is usually the most easily optimized property. And what we struggle with in the academia setting is usually having an active drug. And once we have an active drug, we think that we got for this, and we are almost there. But no, a lot more problems waits for us around. If you want to make a medicine, a pharmaceutical drug, you have to operate in a pharmaceutical context from the very early stage. when you are thinking about the science that you do and are dreaming about a drug, you have to consider all these aspects very early on. Medical, of course, but also legal, financial requirements, so patenting, development, preparing to development in accordance with guidelines of registration agencies. The molecule must be suitable for that structurally, and this is called developability. So very early at the discovery phase, usually at the lead finding, lead optimization stage, you assess developability. What developability is? Developability is the probability of success in development, but it is secured at the discovery phase when you select a proper molecule. If you took care of it and you made some early studies that ensure development later, it most probably go better than if you forgot to do that. If you show all this stuff to your potential investor, it makes the investment much more probable than if you answer, oh no, we haven't studied it. Oh no, we haven't thought about it. So what are the most common errors and deficits that make our molecules not translating to successful drugs? I will show it to you with the help of the stuff that was also collected previously by my fellow colleague from Adamant Pharma, from Adamant Technology branch, which is an investment branch which invests in smaller and bigger projects all over the world, not necessarily pharmaceutical technologies. And there are several areas of problems. First of them is a notion that if I have a drug, that I think that I have a drug candidate because my molecule exerts activity. This is something I previously said. Molecule is active, so why not a drug? First of all, what does active mean? Does IC50 millimolar range indicate activity? Of course not. Micromolar, maybe nanomolar we want. Okay, but I've seen people claiming activity and drug likeness in millimolar range. Okay, but let's consider our drug is much more potent. People focus on activity, especially sometimes in vivo activity or cellular activity, without paying attention to quality aspects. Do I really know what I am evaluating in my assays? People are testing mixtures. Okay, mostly in the natural products world. Not fully understood qualitative and quantitative characteristics. To what specifically activity should be attributed? But okay, we are not testing mixtures. We have one molecule, one structure. Okay, good. Do you have analytical methods for it? And have you produced certificate of analysis? If you are a pharmacologist who collaborate with chemists at the academia and you got some compounds from them and you made some animal studies, how many times you got it with a certificate of analysis? I leave you with this question. Have you seen stability data for this molecule? Chemical stability in different pHs in water, in plasma, in whatever, not mentioning metabolic stability. What is the selectivity of your compound in a therapeutic window? Have you checked it? I've seen a project where people claim they have very potent anti-pain drug, but they forgot to think about respiratory depression or drug dependence, or acting at 150 other targets. Exploring substances which are known pains or toxicophores. What pains are? These are pan-assay interfering substances. For example, people love curcumin or other natural products which because of non-specific interference are claimed to be active in many assays, but they are not. Toxicophores. Okay, I am active at my CNS receptor, but the metabolite of my compound is toxic. Do I know that? Have I checked? Is the route of administration acceptable? Believe me, I've seen projects during which people thought about advancing further to prolonged use compounds which were active only intrathecally and compared them with compounds given orally or intraperitoneally. This cannot happen. Downplaying mechanism of action. Since it exerts activity and it's going to help people, who would really care how does it work? There have been so many drugs, from pharmacology, we know, with unknown mechanism of action. Oh, believe me, the investors care and the regulators care. If you do not know the mechanism of action, that's a huge disadvantage because it means that your data may be simply a coincidence or completely different target than you claim. A dash to in vivo studies. Mea culpa. We also do that or have done that. We have the affinity data at our receptor. What we do, we give it to pharmacologists who inject it to a rat and looks in Porcel test or whatever else test. Shows us active dose. What was it? Was it dissolved? Was it permeable? Was it stable? What concentration this drug reached at the vicinity of receptor? Do we know that at all? Does it have active metabolites or whatever? What are we talking about? Technology-centric thinking. We have to ask ourselves the question, is there a need for yet another drug in this class? Is there space for a novel chemotherapeutic in this indication? Do I bother to learn what unmet medical needs truly are? Have I spoken to a single clinician, or am I living in what I call review paper reality? My colleagues from different countries or different worlds, which are pharmacists, biologists, wrote the review paper or this target, how is it important, and in general, drugs are working through it, and so I'm doing next molecules on this target, and maybe it will be a drug, maybe somebody will be interested in the future. Oh no, I have to prove it. What's my competitive edge over standard of care and novel treatments? Have I compared directly to the best in class compound? And why am I better? Having a 5-HT6 antagonist or 5-HT1A agonist, which is simply another compound which is also active or less active, which can be achievement in itself, is not a competitive edge. Why should it be better? Being 10 times or 100 times more potent than morphine, for example, who cares? What about addiction? That's important. Exploring approaches that has not produced results for decades without clear concept why it's going to be different this time. If we are talking about 5-HT1A agonist, it is only because we have an idea why our compounds are better. Because otherwise, having another 5-HT1A agonist just like this doesn't make any sense because people have tried it for 40 years. And we cannot expect different results by doing the same all the time. This is a very big problem with old targets. The new targets have this novelty reward. Investors love novel targets, but novel targets are in many cases simply not good targets. The older targets are better validated, but we have to have an idea why our compounds should be better, not just because. What is better? Do I have the clear vision of target product profile? Target product profile is a set of properties of this drug product. Population, route of administration, many other features. This is a topic for a different lecture. Faulty experimenting. This is something that sometimes because I previously presented similar presentation and I saw the reaction of people, they were pharmacologists. Faulty experimenting, what are you talking about? We are scientists, we do not faulty experiment. Okay, studies inadequately controlled or not controlled at all. For the last 10 years, I've been serving first as an expert for NCBR, so National Center of Research and Development, and then in adamant technology as the adjacent expert for potential investments. You wouldn't believe how many unbelievable dossiers I've seen. In many cases, people are not doing the experiments properly. The experiments that would probably won't be accepted as a paper are sometimes included in drug discovery dossier for academia and smaller companies. Comparing against each other compounds with different routes of administration. As I said before, one administered intrathecally, another intraperitoneally, or one orally, another intraperitoneally, or mine orally, control intraperitoneally, because I had some historical data and who cares? Testing compounds in different strains, different species, using different methods, different labels. Single run experiments, not repeated, not confirmed. No quality data on substance tested. This is something I mentioned before. I have a powder or something from the chemist. Do I bother what it really is? Have I checked it, or I believe that it's probably okay? Have I seen a certificate of analysis? Insufficiently powered experiments. Unclear methodology. Something that is not mentioned here, but in many cases, the concentrations that are seen in vitro completely do not match the in vivo concentrations that can be obtained given the doses that were given. Okay, so no PK/PD match, suggesting that this is not this target. Sloppy document. I would bet that 90% of academia-based research is inadequately documented considering the requirements or expectations of pharma investors, including ourselves. Taking hypotheses for facts because that's what literature implies. Again, review papers, reality. What are hypotheses? What are facts? Problems with intellectual property rights. No patent analysis before the project launch because we are scientists. This should be done by, I don't know, someone else, maybe legal guys or company who is interested in our brilliant research. They will do that, maybe because they have specialists. No. If you want your project to go further, you have to do it yourself or meet somebody else who will do that. Your academia should help you. But it has been done. Disclosure before filing applications. I've seen a project when professors came with a very nice approach, said that it was not disclosed, and we made patent check and we found 8 publications on this technology. And we asked them, what's going on? And they said, but these are only our publications. Only we published about it. No, nobody else. Disclosure is disclosure. Confusing patent application with granted patent. The fact that you filed a patent application doesn't mean you will get a patent. Overestimating the value of a national patent over the broad worldwide, very narrow claims. believe that structure novelty, which gives patentability, automatically translates to patentability and inferring FTO from patent grants, so that patentability doesn't infer patent purity, okay, freedom to operate. So the fact that your compound is patentable doesn't mean that you are not infringing the other patents. Overprotecting your data, very common situation. We cannot show it because it's classified. Then we enter into confidentiality agreement. We open the dossier and we see that it was overprotected for a reason. Nothing there. Manufacturing. Is it my problem at all? Am I aware that a huge amount of materials is required for tox studies, tox batch, and for clinical trials? Am I aware that different methods of synthesis will be required? Not only additional costs, but time and risk before we get from discovery to development. Is my product fit to be manufactured at industrial scale? Do I use exotic manufacturing methods that are not utilized by CMOs, so contract manufacturing organization side? Rare, expensive building blocks, reagents required, or for biologics expression system available at any CMO globally? What are cost of goods for the synthesis, for example? What shelf life my stability data indicate, under which conditions, time zones, etc. This is complicated, and this is something that we cannot take care about at the early discovery phase, but we have to consider these facts as early as possible. And we have to know that manufacturing issues are a very good reason for a project failure, especially among biologics, the same as lack of efficacy. So, in summary, drug discovery is a multifactorial research and optimization process, okay? Activity is of utmost importance, but is not the only factor. Good insight into clinical context is crucial. Is there a real unmet medical need, or am I just, walking around the area that I've been for 20 years and read a lot of review papers about? Evidence, not assumptions. Is there any idea for competitive edge? How do I differentiate why my molecule, my approach, my mechanism of action, whatever, should be really better than what's the best available? Solid design of experiments and documenting. Probably solid design is proper in most cases, but documentation, probably not. IP protection of utmost importance. You have seen this 20 years of patent protection is especially important in the last several years, during which our successful drug really brings value to the investor. Inability of manufacture of a drug at a reasonable scale and cost is a good reason for project failure, as good as lack of efficacy. So answering the question that I asked in the title, why are all these brilliant molecules don't become drugs? Because the scientists are forgetting the regulatory context. I am a scientist. It will be done later by somebody else. I am working on technology, on science, depending on poorly translatable preclinical models. Sorry guys, in the CNS, it is especially problematic. If you only show data from an animal model and nothing else, it won't probably work, because there were so many failures, so many lacks of transitions to the clinics, that the investors will tell you, very interesting projects, show me the phase 2 data. And good luck in finding money to get there. Ignoring developability of a molecule, okay? So ignoring the features that may decide on the success of development. Focusing on activity too much or only. My molecule works in a lower dose than clozapine. So why isn't it a drug? it's poorly soluble, unstable, and I don't know 50 other things about it. Underestimating the risking of the project. So the risking is looking for studies that I could do to show potential investors that I ticked all the boxes, that I checked everything I could early on that decreases the chance of a failure later on. My molecule is active in 6 memory models, but I don't know anything about the QT prolongation, or I know, or it turns out later that it's arrhythmogenic or impairs motor coordination. So it's useless. And last but not least, unable to secure financing. And I think it's one of the greatest problems. We have a project that has really compelling preclinical data, but its patent is quite old. And we are talking to investors now, and they are really hesitating if they should enter into further talks, because they looked at this life cycle and they are thinking, will be on time with all these clinical trials to, benefit from the patent protection, or is it too old? Scientists sometimes have problem with patents, sometimes are not convincing in showing the unique selling point of their project, or unconvincing, de-risking, poor documentation. These are the most important problems. What are key takeaways? I think 3 things. Focus on a critical path of the project. What's your competitive edge? What's your advantage over the others? And developability. So basic things that may make your molecule not developable have to be identified very early and discarded. De-risk your project as much as possible and know that safety first. There are medicines on the market that hardly work but are safe. They will be accepted. If they pose safety problems, nobody will invest in it or accept it to the market. And collect a clear and thorough documentation, because this is your product. What you then show to your investors or show to regulators is a dossier, not the powder or other stuff, a documentation which is usually poor in academia setting. That's it from my side at this moment.